Okay, so here is our course homepage. Just want to show you where things are going to be so you have an idea of what to look for. Obviously, you've all discovered the Google Meet, otherwise you wouldn't be here. I'm going to put main news stuff in here, so whenever you click onto our classroom, always just take a quick look at course news because there's going to be something there usually. There's also, if you scroll down a bit, on the right-hand side, you've got our calendar, and I've inputted the tentative dates for all of our quizzes and all of the major assignments, everything is in there. So you're welcome to, go to click on calendar and you can go and see what you've got upcoming. So everything's kind of listed there, including where to click on to actually get to the quiz or how to submit something. And you can see we've got a whole lot of stuff, 4, 5, 6, and 7. We got something every day for the next few days. So you'll survive. Don't worry. What else do I want to tell you? So there's this frequently asked questions section up here as well. Nobody's ever used it. This is my third time teaching grade 12 this year in the quadmester system, and nobody's used it yet. But if you do have a question that you think maybe it needs to be not just you wanting to know the answer, but it's useful for everybody, just type it in here. Give me your name so I know who I'm talking to, and then I'll write you back, and there you go. So you can use that if you like. The main goodies are going to be under content, but I'll go there in a second. Also, I have all of our quizzes listed here. So everything upcoming, you can see I've got the whole quadmester sitting here for all your tests and quizzes. Sometimes it's a quiz, sometimes it's an assignment that gets, it's just easier for me to mark it if you submit it like a quiz. But anything that would be like this, like the labs or whatever, those are more open book kind of a thing. Like you're just using whatever you just completed as your work, you're just submitting it to me this way, just because it's easier for marking than Dropbox would be. So a lot of these are tests and quizzes here, full-on tests and quizzes. Please treat every test and quiz like you are never allowed to use anything but your memory. There's very rare times when you'll have a resource. Please, please, please be honest. The temptation to cheat is huge this year. Don't be that person. Please, please, please. Unfortunately, if I do catch that you were cheating, which actually isn't that hard, if I look for it, I can find it. I can see timestamps of every time you guys click a button and test and all that kind of stuff. I can see how long you spent on a question. I can see how long you were away from the computer for, all that kind of stuff. I'm not expecting cheaters, but I'm just saying, do not risk cheating, okay? Please make sure that everything is done honestly. And don't be tempted. Even the most people with the most integrity are still tempted to cheat. But don't give in. You got this. Come out at the end proud of yourself that you actually did what you needed to do. Okay. So tests and quizzes will all be there underneath quizzes. There are a few things to hand into Dropbox. You'll see them as we go. Some of them will be. I guess this one is for our next unit. So none of these apply for this particular first unit. Okay, moving on. Content. Okay, so as soon as you open up content, underneath course docs, the main page here, you do already have your calendar. So please notice this. We've got a little tiny quiz tomorrow. It's 22 true-false questions. It's online. It's meant to be easy. Some of them are even jokes. But that's just to get the safety stuff out of the way, and I'll run through safety shortly. And then you've got a real quiz on Wednesday. You do want to study for this one. Make sure that you're good to go. And then we've got an optional quiz. This is just to give you a heads up for types of questions you'll see on the final test. And then we have another real quiz here as well. Very small. These quizzes are important. I know it's annoying because it looks like, hey, you got a quiz every stinking day. But these are important because they, not so much this one, but these two here, because they give you the types of questions that you can expect on the test, and they take some of the heat off the test. And what we were supposed to have is this being more spread out, right? We were supposed to have this over 7 days, and now we've got it over 5. So unfortunately, it ends up being a little quiz every day. But you should be fine. It should help you get ready for the big test. And you notice that you're going to have the whole morning section before lunch to do your review on Friday. And then you'll have your test in after the lunch period at 10:45. Anyway, that's where we're headed. Please make sure that you do them the day that they are assigned. You won't have very long to sign in, but I'll talk about those individually on the days that we do them. This lab quiz will be open any time between 8 and 2 tomorrow. I don't care when you do it. It doesn't really matter. you're not going to cheat on that one for sure because it's so easy. You'd be silly to lose your integrity over that. But the macromolecules one, super important. Make sure that you're studying for it before you do it. And then same thing with the cell organelles ones. Make sure that you've studied for it so that you can get the highest mark leaving here, okay? So again, I've got week by week. You'll notice that just the way it lands, we seem to have a test every Friday. Test again on this Friday here, when we get back. Hopefully we'll be back together in person by the time we hit May 31st. Who knows? But hopefully this week I'll see your faces. It's going to get really boring for me on this end talking to this computer the whole time. And again, this is our final day here. Now, in our whole quadmester, we have this weird day over here, this one day at the end of June that's supposed to be in our course, but I have to write all your report cards here on the 25th, on the Friday, by 2 o'clock. So nothing can count for marks here. So what I'm going to do is just fun day. I mean, we're going to extract some DNA, so we'll do a little lab, and we'll watch an interesting video, things like that. So it's not, I can't give you any marks for anything here because I will have already finished your report. So just so you know, we've got this weird day. That brings us down to, I think, 20 days. So we've got 20 days to do our course, which is unfortunate because your other course in the week D, if you've got one, is going to be 24 days. So we're really crunching stuff in, but you'll survive. We'll be okay. Okay, so whenever you want to know what's upcoming, just click on course docs, or it'll open up as soon as you hit content, and it's sitting right here. Course outline is here. This is all the boring stuff. You can go through and you can kind of see what kind of topics we're covering. This may have changed a little bit because of quadmester system, but it's roughly the idea. You'll notice the one thing that actually does matter is that when you're looking at your course breakdown. Oh, by the way, hang on one second. When you're looking at this stuff here, when you're seeing everybody's stuff, I don't know if you know this, but you can actually pin somebody so that this becomes the most important thing. So if you wanted to, when I'm presenting, if you want to pin that particular one, you'll be able to see the full note. So I don't know if you guys have all figured that out, but just in case you haven't, you might want to pin it. Go back to this. Okay, so knowledge and understanding is a bigger bulk than normal. In grade 9 and 10, this was all like equally weighted, but in 12U, there's just so much to memorize that the knowledge gets a little bit more heat, so it's about 35% of your mark, and then communication is about 15% of your mark. So that would be like diagrams, being able to express yourself properly, proper spelling, all that kind of stuff is for communication. And then application and thinking are the same weighting, 25 and 25. Okay? More boring stuff. Everyone would have picked up one of these. If you could still fill it out for me, please. You should have a paper copy of it with you. You just have to fill it up to this part. Don't worry about the second page where you've got the attendance record and all that kind of stuff. Don't worry about any of that. Just come up to this part here, so I've got your information. But let's get into our actual biochemistry unit now. So I've got your unit outline posted right here. This is what's on the front page of your big booklet. And the way the colors are meant to be read is that this first green block is meant to be today, day 1. We won't get all of it done, but at least the good chunk of it. Then the white is supposed to be tomorrow. Green again would be Wednesday, and so on. It's meant to be one day at a time kind of a thing. But when I created this, we had 6.5 days. So unfortunately, some things did have to get crunched. And if you go back to course docs, we also have this huge big lab we were supposed to do. We actually have 2 labs we were supposed to do together. So I'm going to have to figure out how to get that done. I've squished them into the same morning so that at least I can talk to you about it, walk you through it. I still got to figure out what I'm going to do because obviously you can't do this stuff at home. So everything's kind of been crunched. So that means that if I go back to biochemistry, that means that this is correct, but it's a little bit out of order now because of the labs are kind of messing stuff up. So the color scheme doesn't work anymore, but don't worry about it. As far as stuff on the side here, I know I'm talking like crazy here, but biochem stuff here. Textbook files will always be the top thing. You can see that every unit, and I've given you the page numbers so you can find where your homework is. Note booklets are here. This is important for those of you who haven't picked them up yet, and for everyone else who has picked them up, but you still need your note. This would be the current blank booklet that I printed for you, C-Quad 4. This is the one that you guys have in your hands, or you can come and pick it up from the main office anytime before 3 or 3:30 if you haven't done that yet. There's only a few of you that didn't have it. If you're repeating the course and you did it just recently, like last quad, it's the same book, but it might be a little different from anyone who's repeating from quad 1. So just make sure you note the differences. And here's where I always put in the completed booklet, which is what I'm going to have to use today because I can't get my iPad to work. So this would be your just the notes kind of a thing. And it just runs through. So what I'm going to do is I'll teach from this. I'll do the bigger version. I'm going to teach from this, and then I'm just going to give you guys time later in the day just to copy it and get down whatever you want from it. But there's a page that's going to be missing in between here, and that's the one that's sitting on my iPad that I can't get to. So I'm not sure what to do about that, but I'll try and fix that for later. So everything will be here. This is the entire unit, and that'll be the same for every time we do a course. Anything you need to write down, the booklet will already be here. Then we've got, let's see here, lesson videos. I didn't do these. This is last quad my teaching last quad. The first 2 I didn't record. I'll have to do that and upload that eventually. So you can come to lesson videos. Obviously the first couple aren't there, but then there is from the last quad, I recorded and I edited out any kids that were talking, I edited out and all kinds of stuff. And I just tried to give you a sample of what the real lesson would look like. So if you miss something or you just wanted to see it again, these are pretty faithful notes to what we've already done. So you can just come back and take a look at them. Hopefully you won't miss any lessons, but it's a resource if you need it. Some of the animations that we'll be looking at are listed here as well. Obviously it says animations. Lab handouts, I'm still trying to figure out what to do with this. Again, I don't know how to do the labs with you guys. I'm still going to figure that out by Thursday. But the idea being, these are pictures from last quad, although some of these results are a bit wonky. But I can walk you through what you should have seen. And then, where are these here? So these are the actual results and stuff that you would have seen, some real pictures of what the lab looked like. So I might just walk you through this and then kind of do like thought experiments. Because this stuff is important, it matters for the test. And it matters for understanding the concepts that I'm teaching you. That's why I love doing the lab in the first week. But unfortunately, can't do anything about that. So everything will be here. We'll talk about it. The results are here. I'll probably put together a little presentation so I can walk through it a little easier than scrolling. So that'll be there for you as well. You've got I think this is tomorrow that you have to do this. There's allosteric enzymes. Any links that you need that's in a handout is listed online. So they're all here for you, little short things to run through. And then a few of the answer keys, not all of the answer keys. Some of the ones are available here are just available in class right now. But then some of the main ones will be listed here. So you can check your work. Please make sure you're checking your work and not copying. It's useless to you if you haven't actually thought it through. So don't waste your time. There's no reason to fill in blanks if you haven't actually thought about the answers. So wrestle with it first, think it through, and then the answers are here for you guys to just quickly check. Okay? If you do get stuck on a few of them, that's not a huge deal, but please don't just copy. It doesn't help you. Oh, and I should say, feel free to click the button so it dings on me, and I can see that you've raised your hand or whatever, and then I'll call on you, and you just ask your question at any time. Don't worry about interrupting. If I'm in the flow of something, it doesn't matter. It'll be just like as if you're here in class. If you want to put up your hand and ask a question, please do that. It helps you to be more engaged, and it helps anybody else that maybe they had the same question, didn't know how to ask it, or were too shy, and you might have just saved somebody. So please make sure you're be brave, ask questions, and we'll get you through this quadmester system as best we can. If our internet cuts out at any point in time, not your internet, but my internet, if I suddenly disappear for a while, I'll obviously keep trying to log back on, but sometimes that can be an hour. You know how it works here at school. If that does happen, then just go and start filling in notes and just start doing homework. And then just keep an ear out, and I'll pop back into the meet as soon as I can. Good old technical difficulties. We're going to do the boring lab safety. This is what the quiz is based on tomorrow. And I want to get this over with right now, so let's do that. So first of all, obviously, you're not doing any labs, but when you get back, and we are in the classroom, we might have a lab or two to do before you guys are done by the end. Still have to go through it. Lab safety doesn't matter whether you're in the building or not. Obviously, follow instructions. If you don't, you sit out the lab. Easy. Read everything in advance. This includes what's going to happen on Thursday. You guys have the little booklets for both of those labs. Read through the labs ahead of time so you have some idea of what I'm talking about. And then when I'm showing you this is where the materials are, this is what's happening. You already have sort of an idea in your mind of what's supposed to happen, so that I'm just clarifying. So read things in advance. That would be wise, okay? And if you don't understand anything, ask me about it, but don't just walk up or talk to me and say, hey, how much am I supposed to get of a particular chemical? No, did you read the lab? Because it said 5 mils. So you read it, you make sure, but if you are stuck, you always ask me and I can help you out, okay? If you had any food allergies or chemical allergies, that's something that your teacher needs to know. If you were in the room, you would know all the safety equipment, how to use the eye wash, all that kind of stuff. You're not here. I'll do that when you guys get back. I'm sure you've seen it before. If there's anything broken, again, you can't break stuff here because you're not here. But if there's anything broken, let me know right away, and then we throw it away in the right way. Everything's got to be labeled when you've got your containers. Can't go into the back room unless I ask you to or tell you to. Don't take any chemicals away with you. Even if you have a nice container for them, you don't take anything home with you. Hint, hint, that's probably one of the true-false questions tomorrow. Never take anything home. When you're working with preserved animals, you treat them with respect. So last year when you guys, hopefully you got to do the pig dissection. If you did, or maybe you did the frog, or maybe you did the eyeball back in grade 10, then obviously you don't run around and treat them like puppets or something. You just, everything with respect. Let me know if there's anything, no matter how minor. This is probably a quiz question. It usually is, because you aren't qualified to know whether something is major or minor. I'm barely qualified to know whether something is major or minor. So even if you just, paper cut or something, even something simple, just let me know. Let me decide whether it's important or not, and let me treat it for you. So please, anything, accident, injury, or emergency, let me know, even if you think it's nothing. Okay. Always wearing protective equipment when necessary. Now, goggles aren't necessary for every lab, but when they are, you need to have them on until we're done. You're always standing up, never sitting down, and that's for 2 good reasons. When you're standing up, you can jump away quicker in case something spills. Plus, if you're sitting down, your lap provides a lot more surface area for things to fall on. So you want to make sure that that isn't an issue. Always tying back long hair, anything that's loose, anything that's dangly. Make sure that you don't have anything that could dip into chemicals or fire. Lab coat and apron when appropriate. We never wear them, so this doesn't even matter. But this does matter. Your shoes have to always cover your feet, so you can't wear sandals. I think most of you guys know that already. Scalpels and knives. We won't be carrying any, but always pointed down with your handles so that if you were to trip or something, you wouldn't stab anybody and you wouldn't stab yourself. Lasers do not go into anybody's eyeballs. And same thing with the sun. You're never looking directly at it. And that's because the intensity is too high. It'll damage the retina at the back of the eyeball permanently. And it's not fun to have that little blind spot in your eye when somebody's done that to you. It doesn't matter even if it's in a reflection. Even if it's in a reflection, it's still going to be just as intense. So you want to make sure you're not looking at anything bright. Okay? Don't eat things in the lab unless I tell you to. So unfortunately, that's different with COVID rules as well, because you have to eat lunch in here. You don't have anywhere else to go. But if there's chemicals and stuff, we just don't eat them unless I tell you to. Glass tubing, I need to take this one out because we don't use that anymore anyway. Never working without me. Keeping everything clean and clear. Again, that's something we'll talk about when we actually have a lab. Cleaning up spills immediately. Don't leave anything there. If you do get something splashed on you like a chemical that could burn you, you want to make sure that you are rinsing 10 to 15 minutes or until professional assistance is obtained. So that would be either under the tap if it's your skin, it's your eyes open in the eye wash station if something went into your eyes, et cetera, et cetera. And we also have the safety shower that I would have shown you if you guys were here, but you're not here. You can, same thing if you're burnt, you're more likely to get burnt at home anyway, but if you're burnt, you want to throw that underneath the water for at least 15 minutes as well. And what that does is prevent some of the injury from being worse than it could be. Cools it down, prevents some of the inflammation, etc., which could cause extra damage. Pipetting is basically like using a big long tube, like a big long straw to suck up chemicals. We used to use our mouths to suck it up like a straw, and we just never do that anymore. Now we use little bulbs like the end of like an eyedropper kind of thing, but this is like a giant eyedropper. You need to waft. That's so if you're smelling your coffee, you don't shove your nose into the cup. Instead, you use your hand to bring the smelly air to you. That's wafting. So you need to waft. Don't pull on the actual cord of a plug, pull on the plug itself. You guys have probably witnessed this with your like charging cords and stuff like that, how they get frayed. So this would have been if you were using the microscopes. You pull out the actual plug itself, not pull on the cord. Do this at home and your devices will last longer. Where are we at? 27, never use broken glassware if it's cracked or broken. We use special safety glass, which is able to withstand higher heat than regular glass can. But if it's cracked or broken, it compromises the glass. And what can happen is when you start to heat it up, because it's now compromised, you can actually shatter. So we would never use it if it's cracked or broken, and we just put it, you just toss it. If you pull it out of the cupboard and it's cracked or broken, you toss it. If you crack or break it, you still toss it. Bunsen burners, we don't actually use these in our course, but you would in other courses, so we still have to do safety in the lab. Never leave a flame unattended is the basic thing here, and turn off the gas when you leave. Retort stands, those are those fancy stands that you can like put clamps on, so you can lift up like a ring clamp or something that you can lift up and put the like a beaker on so that it can go above the hot flame. It just means that make sure that they're not wobbly and make sure that they're level. Hot items on the lab station. Most of our labs in the building are actually okay with that because they're metal. Here in the bio lab, we actually have like a laminate countertop. So if there's something hot we don't want to burn, we don't usually use anything hot enough to burn, but if there was something hot enough to burn, we would put it onto like a ceramic tile or something like that rather than putting it on there. And obviously, don't touch it with your hands if it's hot. You use the right type of tongs or clamps. Read everything first. Don't use anything you're not supposed to use. Scupula, hopefully you know what that is. I'll just find a little picture for you in case you've never seen a scupula before. Okay, this is a scupula. So you've seen them in labs before, hopefully. So it's just a little metal, like a spoon kind of a thing that we use, a little spatula spoon. And basically the rule is just don't mix them. One scupula per chemical. Otherwise, if you dip this scupula into a different chemical, you're going to contaminate stuff. That's no good. So that's very basic. Again, I know some of this stuff is like, oh my gosh, this is so boring. Why are we doing this? We have to do this. This is part of the requirements. So I'm stuck doing it. Feel pity for me. You have to hear it once. I have to do this every time I teach the course. Okay, so don't mix anything you're not supposed to. Make sure everything is tightened up after you're done using it so that we don't lose stuff. This is especially important for things like hydrogen peroxide. Hydrogen peroxide will decompose naturally into water and oxygen if you don't keep it sealed and if you put it in the light. So make sure you're tightening up these containers when you walk away, even if someone's right behind you to use it. When you're heating your test tubes, you're always pointing away from yourself and others. So if you can imagine the lab bench, okay, just follow my mouse here. So there's a box. You see the box here? Pretending that there's a box here. Hopefully you can see that in your imagination. Your box is here. This writing here would be considered the wall, and then your lab station comes out here. You can have a person on this side. You can have a person here facing the wall, and you can have a person over here. So if you've got a heated test tube, when you heat stuff, you can cause chemical reactions, you can cause steam to come out, you can cause all sorts of things to be explosive depending on what you're heating. So you always want to make sure that the open mouth of the test tube is pointed towards the wall, not to any partners or to anybody at lab stations nearby. Simple enough. Okay? And the same thing, if it's heated, don't look at it from the top of the test tube because you have the same danger. You want to make sure that you're looking at it from the side if it's heated. Not so dangerous if it's cool. Okay? This one, definitely on the quiz, it always is. When you dilute acids, you always pour the acid into the water. So you always add the acid. I want you to imagine, we'll go on a little imagination journey here. Imagine that you are standing out on a dock and you've got yourself a nice flat rock and you're going to try to skip it across the lake. And if you suck at it like me, when you attempt to skip it, it's not going to skip at all. Instead, it's just going to plop down into the water. And you can see the rock kind of floating down. But when the rock first made contact with the water, with the surface of the water, the rock itself compressed the water, and it kept going down because of its density, but the water splashed back up, right? Can you picture that in your mind? You've got the rock going down, smacking the water, the rock continues to fall, and then the water splashes up. Well, now consider that you've got two liquids that you're trying to mix together, okay? You're trying to get a diluted acid, so you've got a strong acid, and you've got water to dilute it. Treat the acid here in your mind like the rock, and then the water like the water out on the lake. What would you rather have splashing back at you? Would you rather have acid splashing back, or would you rather have water splashing back? I would rather have water or very dilute acid splashing back. So the thing that you are pouring, or the thing that you are adding, the acid, even though it's a liquid, it'll compress the water and the water would bounce back. If you added the water to the acid, the water would compress the acid and the acid would bounce back. So I don't want that. So I'm always going to add the acid. AAA. It's a nice little alliteration. Always add acid. The other reason for that, not only because that's what you want splashing back would be the nice diluted water, but the other thing is that if you are imagine you're holding a beaker full of the acid, you control your flow rate. You can pour more or you can pour less, whereas you don't control what the water does. The water's just sitting there. You have no way to stop it from splashing other than how fast you're pouring the acid. So you want to be in full control of the acid the whole time, and you want to make sure that it's not the acid that splashes back, it's the water that splashes back. So you always pour the acid into the water, always add acid. That's definitely a little quiz question. So always add acid, make sure you got that the right way around. This is so important, and I see kids do this wrong every year, even though I tell them over and over and over again. Never, ever, ever. Once you take chemicals out of the main original container, never put them back. So once you take it, you either have to use it or you throw it away. Please never put stuff back because you're going to end up contaminating the original container. It's the same idea where, let's say you share a drink with a friend. You don't want them to take a sip of your drink and then they, spit the rest of it back because they didn't need as big of a drink as they thought. Like, that's just backwash. Don't do that. So same thing here. Don't backwash into our original containers. That's gross. And you contaminate the entire container, just like your friend spitting back into your drink would contaminate your drink. It's very gross. Please don't do it. This says never dispose of chemicals down the drain. That's a big fat lie. There's a lot of things we do dispose down the drain, but there are some things that there has to be a separate waste because we can't put it into the municipal water. So that'll be on a case-by-case basis. And then make sure that you're using the right size and shape. So if you're carrying 200 mils, you don't want a 250 mil beaker. It'll be too close to the top, so you'll get yourself a 400 mil beaker. So that way it's nice and safe. You're not going to spill anything. And then always make sure you're at your experiment till it's finished. At the end of the experiment, make sure your work area is clean and dry, equipment's put away, wash and dry your hands at the end of the experiment. So that's our little safety conversation. Again, there's a little quiz tomorrow. You can do it anytime between 8 and 2. I'll remind you tomorrow when I see you then. And it's based on these rules and based on common sense as well. Okay, like if there's a fire, stop, drop, and roll, that kind of thing. Okay. This sheet here, as we go through biology, you're going to find that we're using a lot of these very scientific words, but really all they are is little sentences in a word, because you're going to use these prefixes and these suffixes. Prefix means before the word. Suffix means at the end of the word, but they can actually be interchangeable in some cases. Sometimes you'll get a prefix used at the end. This is just a small sample of a lot of the Greek and Latin words that we end up using, but you can put words together using these little pieces. So I can show you a word you've never seen before, like something like periosteum. And if you know that peri means around, like perimeter, and then osteum, anytime you see osteo, it means bone, then you know if someone mentions the periosteum that they're talking about something that's around the bones, things like that. If you see this here and you say, okay, what is a hypodermic needle? Well, hypo means to go under or to be below, and then derm is your skin. So if you've got hypodermic, it means it's going below the skin. We'll see the word hypo and hyper, which is its opposite, which means more. Lots will be using these words. So if you know the little pieces of language, I can throw a brand new word at you and you can pretty much guess within reasonable distance, guess its meaning. There is an answer key. I haven't posted it yet, but maybe I'll scan that over lunch and post it for you guys so you can check. But I encourage you to go through and do these, because if you can start to get the little pieces of language and you kind of understand how words are put together, you're going to feel better about all these words I'm throwing at you this year, okay? Tons and tons and tons of terminology. This here, the Chemicals of Life worksheet, this is an ongoing worksheet. It's not meant to all be done in one day. It's meant to be a little bit at a time. So we're going to be talking about our major topics. We'll talk about the macromolecules, which would be like actually let me scroll up to the top here. I'll show you this is the old unit outline, but it's got the same kind of idea. So today we're supposed to be talking about this organic chemistry stuff, which unfortunately is the page that I can't get off my iPad. So I'm not sure how I'm going to do that, but we'll figure that out. Then you got carbohydrates and lipids and nucleic acids and proteins. These guys here are the macromolecules. There'll be a quiz on that on Wednesday. We're going to talk about their structure, talk about their function within the body, and all that kind of stuff. Last year in grade 11, you would have talked about how to break these things down in the digestive system. Now we're going to talk about how to build them. Once they're broken down and absorbed into the bloodstream, how does your body build them? We'll talk about that this year. We'll talk about enzymes and how they work. And then we'll also talk about cell organelles. But mainly, we want to focus on the cell membrane this year. That's the main important thing. Up until now, we've sort of treated the cell membrane like as if it's just this container. But it's actually a really amazing organelle that kind of gets skimmed over in grade 9 and 10. So, or grade 8 and 10, I should say. So cell membrane, super important, really amazing. And then we'll talk about passive and active transport, which is how the cell membrane transports stuff into and out of the cell. This unit is massively foundational for the rest of our course. We're going to keep talking about the macromolecules. We'll keep talking about enzymes. We'll keep talking about the cell membrane, and very much so passive and active transport right to the end of our last unit. So it's really important that you do well on the first unit. And if you have any questions, you need to be asking me as we go, because this literally is your foundation for the rest of the time. Okay. All right. Some more boring stuff. Let's get through. So please make sure you come back and do this little thing. But this Chemicals of Life worksheet is meant to be for the first unit. That's why it's in the first unit book. But we're going to do a bit at a time. So over the next couple of days when we're doing all of the macromolecule stuff, you can come back in and do macromolecules. Now, some things like globular protein and fibrous protein, these might not be necessarily straight from our notes, but they might be in the text. It's actually not that difficult. They're both types of protein. A glob is just like a round bundle. And fibrous just means long and thin. So that's all. So that'll help you out for some of those questions there, but we just highlight that. So come through, do a bit at a time, and there is an answer key for this one already posted online. And then these questions as well kind of give you a heads up for what's going to be on the test as well, on the quiz and on the test. So if you get this stuff done before the quiz on Wednesday, that's good. You're on task for that, but it doesn't have to all be done today because I'm not going to teach it all today.